Hello and welcome to our fifth video on molecular biology topic 2, and in this video we're going to be looking at enzymes. Okay, so you would have been introduced to enzymes way back in year 8, and I'm sure you've looked at them since then, hopefully. But what enzymes are is they're proteins. Now we know what proteins are. We've looked at proteins in the last unit, but now we're building on that. Okay, so enzymes are proteins that speed up the rate of a chemical reaction without actually going under any change themselves. So in the diagram here, we've got a starch molecule, so our polysaccharide. It's a long chain, it's linear, so it could be cellulose, it could be amylose. Now I can tell you this is amylose because the specific enzyme working on it is amylase. Okay, enzymes generally end in ASE. Okay, so here we've got amylase breaking down amylose. Okay, and what you'll see is the enzyme fits like a lock and key onto our chain of polysaccharides there, and what it does is it breaks down that chain of polysaccharide, in this case into disaccharide units, which is our maltose here. Now this is a very this is just for your understanding. Enzymes do not look like green blobs like that. Alrighty, so before any reaction will actually occur, in most cases we need what we call some activation energy, because we may have the reactants here, for example A and B, but for them to actually react we need some input of energy. Some reactions don't need an input of energy, but a majority do. Okay, so I want you to think way down to the molecular and atomic structure. Okay, and molecules within the cell, all molecules, but let's think within the cell, we're in biology, are constantly moving and colliding. Now, chemical reactions only occur between molecules when they receive enough energy for those collisions to occur. So if the molecules haven't got much energy, they don't move with as much force, and there's less chance of molecules and atoms colliding. Okay, a reaction only occurs if collisions occur. So if you increase the energy to those molecules, you increase the amount of collisions, you increase the amount of reactions. Now the amount of energy needed to break the reactants bonds, okay, so imagine they may bump into each other, but they need to do so in enough force to break the bonds. Now this is what's called activation energy. Once the bonds in the initial molecules are broken, then new bonds can form and you can get new products. Okay, so imagine you've got reactant A, reactant B, they had enough energy that when they collided, the bonds in reactant A and those bonds in reactant B broke, and therefore they could then rebond, maybe in different structures, to form products C and D. Alright, so we have a few factors that can increase the rate of reaction, so can increase the amount of collisions that will occur. One of these is temperature. So that increase in activation energy is through temperature. So we're increasing the movement of the kinetic energy that the molecules have by increasing the temperature. And you can see this graph here showing the rate of reaction up the side, so up the vertical axis, and the temperature along the horizontal axis. And as the temperature increases, the rate of reaction increases until that optimum temperature, and then the rate of reaction starts to decrease. Concentration of the reactants can also increase the rate of reaction. So if you've got more molecules of the reactants in a set space, there's more chance of them colliding. So here we've got three different concentrations of our substrate, so of our reactants, and we see our rate of reaction there. You'll see that it does actually plateau, so it's not a case of keep putting more and more in. There is an optimal rate there as well. Okay, you can increase the rate of reaction by increasing the pressure on the molecules. By increasing the pressure, you therefore increase the kinetic energy. More reactions can take place. You're removing the space between the molecules, so there's more chance of them hitting. By increasing the pressure, they're increasing that energy, so they're also moving faster. Again, another graph showing how that affects that rate of reaction. Conversion rate here being the rate of reaction. Okay, you can add a catalyst to a reaction. Okay, now by adding a catalyst, your reaction becomes more efficient, basically. So if you look at the graph, you've got the activation energy required without the catalyst, that's that blue line, and you see that optimum rate is significantly reduced there if a catalyst is added. So the energy requirement is significantly reduced with the catalyst being present. Now, as I said, enzymes are proteins that speed up the rate of a chemical reaction without undergoing any change. So an enzyme is a type of catalyst. Enzymes, let's learn about them. They are proteins with a specific function. So we've already looked at our proteins, and we said that our polypeptide chain became the protein in that quaternary phase. Okay. Now, enzymes are involved in almost all biochemical reactions in living things, so they are a big deal in living organisms. They lower the activation energy that's required for a chemical reaction. So as we saw on that slide with the catalyst, okay, enzymes are biological catalysts. They lower that requirement of that activation energy. They are specific to particular substrates. So enzymes will only work on their specific molecule. Okay, so like we saw in those slides earlier, and I said we had amylose and we had the enzyme amylase. Amylase will only break down amylose. Okay, it won't break down a protein, for example. Now, the shape of their active site determines which substrate they will interact with. And if you look on the diagrams to the left, you'll see this is the active site in the enzyme here. So this is a kind of representation where we're more looking at the real thing. Okay. As you can see, only molecules that are that shape will fit into that active site. Okay. Now, without enzymes, the metabolism would be so slow, okay, at the normal body temperature and pH that insufficient energy would actually be available to maintain life. So it's really important that we have enzymes to maintain life. Let's put it this way, without enzymes, there would be no life. So as we've said, they're highly specific molecules. They have that highly specific shape. That active site will only fit specific substrates. Now, a small part of the enzyme, the active site, has the correct shape or conformation to bind with a specific substrate. So that's what we've just said. Now, remember, we talked about these 3D shapes, okay? And it's really, really vital that you understand that an enzyme is a 3D shape. We're not just talking 2D. Okay, and the 3D shape of the enzyme is determined by that folding of the protein and those characteristics of the amino acids. So remember, we said the shape of the protein depended on the order of amino acids as to how those hydrogen bonds would make that polypeptide chain twist and form that globular shape. And then depending on that globular shape, the tertiary structure then depended what extra proteins would attach to our first protein, making that quaternary structure. So that 3D shape of the enzyme is so important in terms of its function and its structure. Now, we looked at denaturing proteins. Enzymes are proteins, which means the things that denature a protein denature an enzyme. Okay, so knowing what we know about enzymes, what do you think are the things capable of denaturing our enzymes? We've already spoken when we looked at proteins about heat and pH being the things that do denature enzymes. We also have heavy metals, okay? What might be a consequence of denaturing enzymes in living things? So if you've got a living organism and the enzymes start to denature, then processes of the metabolism will not occur. That is the end of that living thing. Here we go. So picture that you've seen before in our proteins, okay, and the things that can cause the denaturing of enzymes is that increased temperature, changes in pH, and heavy metals. Okay, so looking at temperature and enzyme activity. In humans, the optimal temperature for most enzymes is 37 degrees. So when you hear body temperature, the reason our body temperature is the temperature it is because that is the optimal temperature for a majority of the enzymes within our body to actually work at their optimal rate. Raising the temperature increases the movement of molecules, which increases the rate of enzyme activity. However, if you increase that temperature too high, our enzyme will denature. And we don't want denaturing enzymes. Okay, so overheating permanently will cause a change in that structure of the protein. It is a change in the structure of the protein, remember, because the hydrogen bonds have broken. However, it doesn't change the structure of the polypeptide chain, remember? Okay, that change in structure of our protein is called denaturing. Once denatured, enzymes will never work again. Enzymes are inactivated at low temperatures. So it's not saying they're denatured, they just become inactive. Inactivated enzymes can become active again, okay? So if the temperature drops, the enzymes won't do their jobs, but when the temperature returns to normal, they will resume normal enzyme function. Okay, pH and enzyme activity. Each enzyme has an optimal pH, and that is specific to the enzyme. So pH is a measure of the hydrogen and hydroxide ions in the solution. A low pH, we call acidic, has many free hydrogen ions, whereas a high pH, which we call basic or alkaline, has many free hydroxide ions. Water is neutral, so around pH 7. In humans, pH is generally around 7.6. However, different regions may have different pHs. Okay, so if you think in the stomach, you'll be looking at a highly acidic environment. Different enzymes have different optimal pH ranges. So, for example, stomach pepsin that we've got here has an optimal pH range of about 2.5. 2.5 pH 2.5, sorry. Whereas trypsin in the intestines, that has an optimal pH of 8. So it just depends on the role of the enzyme. Outside its optimal range, the rate of reaction will significantly be reduced. So what does pH and enzyme activity have in common? If we've got an excess of hydrogen ions, it can lead to bonding between the hydrogen ions and the negative charge on the active site or other parts of the enzyme, and that changes the shape of the active site. So this can inhibit or prevent the enzyme actually reacting. A similar effect occurs with the hydroxide ions, and the change in pH changes the shape of the enzyme so it can no longer bind to the substrate, hence it is denatured. Other factors that can affect this affect the rate of enzyme activity include our substrate concentrations. We talked about that earlier. If we increase the substrate concentration, we increase the rate of reaction until all the active sites on the enzymes are full. Okay, we've then got what we call enzyme saturation. We can increase the enzyme concentration. This will increase the rate of reaction until all the substrates are used up. Working through our list there. Okay, so cast your mind back to our work on carbohydrates, and we are going to look here at lactose. And lactose is a disaccharide produced from the condensation reaction between galactose and glucose. Okay, now there is a condition called lactose intolerance. And this has something to do with the enzyme lactase. So you see that? Lactase, breaking down a lactose. Now, all young mammals are fed milk containing the sugar lactose. This sugar is digested by the enzyme lactase in the liver, and it's broken down into glucose and galactose. Glucose and galactose are small enough to be absorbed into the body. Now, some people cannot make the enzyme lactase and therefore are lactose intolerant because they cannot break down the lactose. Lactase can be obtained commercially from yeast grown in milk. This lactase can be used to make milk that is lactose-free. Lactose-free milk can be consumed by people who are lactose intolerant. So you see how there was an issue and scientific research has found a solution to that issue. This milk contains glucose and galactose, so it is much sweeter and more soluble, meaning that this milk requires the addition of less sugar. Lactase enzymes can also be commercially bought for consumption, so you could take the actual enzyme to help you break down lactose. Quite often you'll see people with lactose intolerance, if they know they've just eaten or about to eat something that contains lactose, they'll take a tablet, usually after they've eaten it. When they think, oh no, that had milk in it, they'll take a tablet. And it stops the nasty side effects of lactose intolerance that could make you require the toilet rather rapidly. Okay, so we have looked at those sections. In class, we're also going to look at this idea of experimental design. I hope you enjoyed your video on enzymes.